Hello everybody, and welcome back to our genetics lecture series. Once again, my name is Drew Wico, and this series has been created to educate the communities of the Partnership for People with Disabilities and Center for Family Involvement, particularly about vital genetic concepts that relate to many genetic disorders. As always, we would love to hear your input on this series, as well as hear any questions that you might have about this material. We know it's a bit overwhelming, so we want to answer any questions that pop up. Today's lecture will focus on the differences in genetic disorders and the challenges that researchers face and continually face connected to these differences. First, let's review last lecture's key terms. Last lecture focused on reading DNA and mutations that commonly happen. Remember that we read DNA in a 5' to 3' direction, and it takes a couple steps to go from DNA to a protein. We need to go from DNA to RNA, then chunk the bases into groups of 3 to get our codons. Each codon instructs the cell on what part of the protein to put next, putting together a long list of directions until the whole protein is built. This whole process is like our ordering of coffee for the entire office. The issue is we speak German, need the order translated to English, then the important information taken from the order. We then talked about mutations, of which there are two major groups: point mutations and frameshift mutations. Below are the 6 specific types that we talked about, but the key details that you need to remember are that point mutations change one base, while frameshifts add or remove bases to shift the entire sequence of codons. Usually, frameshift mutations are more impactful due to the fact that they shift the frame of reading, affecting any codons past where the mutation happens. Today's material is going to start by focusing on the different types of genetic disorders, particularly single-gene and multi-gene disorders. First, we're looking at disorders linked to one gene, also called monogenic disorders or even Mendelian genetic disorders. This second name comes from the scientist who developed Punnett squares and was a trailblazer in the field of genetics. But if we want to do a history lesson, we can save that for another lecture. The example we're going to talk about with this is with cystic fibrosis, which might also be referred to as CF. This is a genetic disorder that follows a recessive inheritance. Remember that this means an individual must have 2 recessive alleles to have the disorder, and individuals with just one recessive allele are called carriers for this disorder. Individuals affected by CF have a mutation in the CFTR gene, a gene that is important for keeping the balance of ions in mucous tissues. When this protein is mutated and made non-functional, their mucus becomes thickened due to the imbalance of ions that I mentioned, causing mucus to stay put in organs just like the lungs. This disorder can do a ton of damage to lungs, especially since it increases a person's risk for pneumonia. You can see on the right an example of the difference in the lungs between somebody affected by CF compared to somebody who isn't. The airways are unable to move the mucus, and that allows bacteria to grow within the lungs. One of the most prominent mutations known in the CFTR gene is a deletion of the 508th codon. Remember that codons are the instructions for how to put the protein together, and a deletion can do a lot of damage since it could frameshift the entire end of the protein. This disorder, as a whole, is a great example for a monogenic disorder, since CF is effectively always traced back to a mutation in this protein. However, this is not the same for all disorders. For disorders that are caused by multiple genes, you might hear these referred to as multigenic, polygenic, or even multifactorial disorders. We will be looking at autism spectrum disorder, or ASD. Autism is a fairly well-known disorder, especially as doctors have gotten much better at diagnosing it throughout the past couple of decades. If we were to try and point to one specific cause of autism, what would we find? According to the National Institutes of Health, or NIH, that isn't necessarily an easy thing to do. Autism is believed to not be tied to just one gene, but thousands of genes. The impacts of many of these genes are not necessarily confirmed, but we have reasonable suspicion to believe that they all do play a part. We do, however, see some alleles or mutations that are a bit more common within autism. One example is a variant form of the gene ADNP, which is found in around 3% of all cases of autism. Comparing this to our previous example with CFTR in cystic fibrosis, how does that account for almost 100% of the cases in that disorder, but ADNP only accounts for around 3% of all cases? Let's look at these two classes of genetic disorders in a slightly different way. Here, we are comparing monogenic and polygenic traits, similar to how we would be comparing monogenic and polygenic disorders. You can see in the left, in the monogenic example, that there is only one gene responsible for the trait. This would result in you either having the trait or not having the trait. Obviously, this specific example would be a dominant trait since there are only 2 possible outcomes. But even if it was recessive, there would only be 3 possible outcomes. This example with the hairline is actually an accurate one since that is a monogenic dominant trait. Meanwhile, on the right, the polygenic example exists for skin color or skin pigmentation. There's not total agreement on how many genes are involved with skin color, but many scientists estimate that there are around 100 genes involved. Well, if that many genes are involved, how do we make that into useful information? Imagine all of your alleles were recessive alleles. That would be one extreme end of this graph. Then, imagine all of your alleles were dominant. That would be the other extreme end of this graph. As we add more genes to this, there become many more outcomes, providing the variation that we see in skin colors throughout the world. However, it all comes back to just how many alleles you have in the genes responsible. This accurately mimics polygenic disorders with the sliding scale of how many alleles influence this disorder being what changes. That is why we see different levels of severity for so many polygenic disorders, and scientists continue to try and uncover what causes these genetic changes. These examples illustrate the issues with doing research in genetics. According to the World Health Organization, or WHO, scientists estimate that around 10,000 diseases are known to be monogenic, but there are an immeasurable amount of polygenic disorders. Many scientists study multifactorial and polygenic disorders because they're such a mystery still, and they more heavily impact how our economy and social structure functions. Even with this, it still comes down to the issue of having too many cooks in the kitchen. When doing research, you need to isolate every single variable and figure out which change is responsible for which outcome. How do you do that when you're working with 50 or 100 variables? We'll look at this through an example. Here are 2 groups of pictures. If I were to ask you what each side had in common, what would you tell me? For the left, you're probably going to say something like, oh, those are all pictures of the Empire State Building, and you would be exactly right. This is like a monogenic disorder, because regardless of all the changes in a person, or in these images, their CFTR protein being mutated is the one thing that they all have in common. But what about the pictures on the right? You could say that the commonality between them all is that the buildings look kind of similar. But is that the only thing in common? What about the exact type of architectural style? What about the weather? What about each of these being chapels or having bell towers? The issue is you can't isolate the exact thing that they all have in common unless you know exactly what I searched when I made the collage. We sadly can't read into what mutations are doing because they don't have an exact thought process to follow. This is kind of how it is with multifactorial disorders. There are so many variables to consider and focus on that we might spend time researching and putting money into one research thought process and going down one avenue, but that ends up not being the answer. It's a massive challenge to research these kinds of disorders, but given that they more heavily impact us and our world, we want to find the answers. Research has a lot more steps involved in it than many people realize. I mentioned how scientists are researching every variable possible with disorders and how much time and effort that takes, but here's a graphic to help illustrate this a bit better. You can see that we have 3 groups in the left section before we reach clinical trials, then 3 groups of clinical trials, and finally FDA review. It seems like such a clean-cut process in this image, but what really happens? Well, the first 3 groups? That section represents years of research, and sometimes even entire careers of research, trying to understand how a disorder works. What about the clinical trials? This graphic makes them seem so simple, but doesn't show that getting through clinical trials takes multiple years just for one drug. This graphic also doesn't show that FDA review of a drug can typically last almost a full year. Overall, if you put the timeline together, the development of one drug can take years, if not decades. It might seem like science is blazing through a trail of findings, but the level of understanding from a discovery of some outcome to a human-ready drug is a large gap to get across. Although it takes this long, scientists want to ensure that they are being safe, so this is a necessary process. The goal is to provide options to help remove any negative factors in disorders while ensuring that they are safe, but that takes many years and multiple efforts before it comes to fruition. Today, we talked about two groups of genetic disorders, monogenic and multifactorial disorders. Remember that the main difference is the amount of genes that are involved in causing this disorder. Each of these kinds of disorders present different challenges in research, but the hope is to learn the underlying cause of each so we can better understand how to remove the negative aspects of them. If you have any questions about today's specific material, please leave questions below. If you would like more discussions about research or ethics faced in the field, please leave a like on this video. We would also love to hear your thoughts on this lecture series and begin to see what requests the community has for topics. Next lecture, we are going to discuss direct-to-consumer testing, just like 23andMe. These kinds of products have become a lot more popular in recent years, so we feel like it's a really good time to discuss the ins and outs of them. On this slide, you will also see a list of other lecture ideas. We currently have a plan of different lectures for the remainder of this particular series, but we also want to see what exactly the community is interested in. If you see a topic in this list that you would love to hear about, or even a topic not on this list, please leave it in the comments below. The more feedback we get, the better we can cater future lecture series. We're planning to do one fun lecture focusing on genetics and pop culture to discuss science fiction, like Jurassic Park for instance, so I can at least guarantee that will be one lecture. Regardless, any feedback on future lectures or how we can improve this series is definitely welcomed. We look forward to hearing back from you and hope you enjoyed. Thank you so much for watching.